Hello there. Welcome to the World of Sustainability, where your academic needs are met. This is another aspect of my lecture series. This is basically for biochemistry students, like my biochemistry people. So this topic today is on DNA replication in prokaryotes. DNA replication in prokaryotes. So this is an interesting part because I say it's interesting because a lot of people have issues with this part and other parts related to this topic. But today we'll look at it in a very interesting light, you understand, to make it easy for you to assimilate other deeper parts of this topic. So let's go right into it. So I told you, like, the way I do my lecture process, I make sure I explain the words very well so that you can understand what the topic is about. So DNA replication means how DNA can make copies of itself. Then prokaryotes. What are prokaryotes? Prokaryotes are simply living organisms that are bacteria. They do not possess a well-defined nucleus, and so on and so forth. So they are simply not eukaryotes. Simple. You understand? Very, very. That's a simple version of what the topic is about. You understand? So how does DNA? How do bacteria copy their DNA? Obviously, DNA in bacteria is a bacterial DNA or bacterial chromosome. So let's go right into it. So a bacterial cell with a circular DNA, bacterial DNA, circular DNA. This is how it looks like. This is the bacterial cell wall. This is the plasmid DNA. This is the circular bacterial DNA. Call it chromosome 2, but to be more specific, it is the bacterial circular DNA. So how does this whole process work of does DNA replication? Each time you hear of DNA replication, I told you how the DNA replication means how it copies itself. So this process has been divided into 3 steps. They include the initiation, elongation, and termination. To easily understand and put it in your head, initiation means to start, elongation means to copy, then termination means to finish. Very, very important. Not too much talk, not too much story. Initiation means to start something, to initiate means to start. Then elongation means to copy, to make it longer. You understand? Then termination means to finalize the process. So very, very important. Based on this foundation we have laid, we can continue. So DNA replication is an important and very interesting part because it forms the basis of different advances in biochemistry as well as molecular biology. So we have to know this well to be able to understand those areas of biochemistry. Then DNA replication in prokaryotes is very simple. Just remember that a parent DNA or the parent DNA has the origin. You understand? That means the mama or the papa of the DNA, the bacterial DNA. Simply put, the 9 be these 2. You understand? So every DNA, both for eukaryotes, prokaryotes, for them to be able to undergo DNA replication, there must be an initiator, a point where the whole process must start. This is called the initiator. Very, very important. Or the origin of replication. You call it the OriC. You know, so you may see OriC, you may see initiator, you may see that point where the whole process will start. Is called the initiator. Here in this case is usually DNA. You understand? DNA. Understand? DNA is like it's a protein that goes to that origin of replication. That's a sequence of DNA where it goes there to identify, okay, this is where I'm supposed to start the process of replication. You understand? So very, very important. Then the next one is that's when you the squad comes to carry out the process. The squad now here includes the Helicase, Polymerase. You understand? So Helicase, they have all these proteins, they have their functions here. The Helicase in this case is a protein that helps to break the bond that holds the 2 strands together. The bond that holds the 2 strands together is called the hydrogen bond. What this Helicase does is like is an unzipper. It will unzip that bond that holds the 2 strands of DNA together. That's what it does. So as it does it, like every spring, that if you try to separate 2 springs, they will look for how to come back together. So what holds them apart during the process of replication is on that protein again. It's not here. It's called the SSB protein. They call it SSB protein, or we call it single strand binding protein. It helps to hold the strands apart while this whole process of replication happens. You understand? So the main guy here that comes to do this work is called the polymerase, DNA polymerase. You understand? That's the guy that actually copies the parent DNA to form a daughter DNA. Very, very important. You understand? So at the end of every process of replication, what is formed is you have what is called the daughter DNA. You understand? There are obviously two of them from this whole process of replication. From one becomes two. That's the idea. Then moving on, and this is a very simplified version of the explanation. You understand? But there will be other videos to make this whole process go deeper into this process of DNA replication. This is just a basis. You understand? So very, very important. Then this is just like to still buttress the points. The whole process of DNA replication. I told you there must be every DNA must get original replication, or it must get the OriC. And there the whole process of replication will still start. Then after that one, don't they there, you don't identify that point, so they will take start the process of replication. You can't form this they call, I didn't mention this before, it forms what is called the replication bubble. You understand? It's like a ball. As it is separating, a ball is formed, because you are trying to make new copies from the old or the parent DNA. Very, very important. Then moving on, you look at it now, you see that there are two strands there, which are the new DNA strands that are being formed from the old. They don't call them the old, they call them the parent DNA strand. So the result of all these processes is what I told you before, the daughter DNA strands. You understand? So very, very important. And each of them must always have their OriC, original replication. Thank you very much. So like I do in every lecture I provide or I make, every lecture video I make, I must always provide you with a take-home question or a take-home assignment that you can try to look into after you have learned what I've said so far. You understand? This is like an assignment, so go and check it. Okay, if from what you have gathered so far. So this part is based on the next lecture that I will make on this topic. Now, this is the question. Explain why a lagging strand must be synthesized in short fragments called Okazaki fragments rather than continuously like the leading strand. I repeat the question again. Explain why the lagging strand must be synthesized in short fragments, Okazaki fragments, rather than continuously like the leading strand. So I will leave you to go do your little research. If you have your answer, you can put them in your comments. I'll do a different video to explain this special part, this special area. Like I said before, this is an introductory aspect of this lecture. So I'll do a video to actually explain this part in full. So thank you very much. I miss us in it. Don't forget to like, don't forget to subscribe, share to your friends. Thank you very much.